talking about everything from standardization to analytical practices and so forth that are recommended as the field grows in size and continues to develop. No, that's not what I want. Hello everyone, thank you for joining us for the short course being offered this year for the Pacific Northwest SETAC annual meeting. Today we'll be talking about microplastics and getting into best practices for robust microplastic method development and detection. So this will be a course that talks about everything from identifying microplastics in your samples to classifying them by shape and size and type. And also we'll get into some QA/QC, so things like making sure you collect blanks to represent any contamination that might be in the space that you're working in, as well as analytical methods. So let's get started. So first we're going to talk a bit about microplastics basics. And this course is really designed for people who are maybe just getting into the field of microplastics, but you may also learn something if you've already been working on, say, samples for a little while and just need to sort of brush up on some of the basics, or to get an idea of where the field stands on a particular question or issue, as a lot of labs move towards trying to standardize their protocols so results, so findings from different labs are comparable to one another. So what are microplastics? And a lot of you might already know the answer to this question, but in some ways it's a little bit more complicated than one would assume coming from outside the field. We're not talking about just one pollutant type, and I think that's the most important thing to emphasize here. A microplastic is potentially, hundreds of different types of contaminants. They come in different shapes and different sizes, different polymers, with different chemical additives or sometimes even sorbed pollutants. You can see in the pictures here, this is from a paper from Chelsea Rockman's lab that was published in 2019, and the famous quote from that paper is, "A microplastic is not a microplastic is not a microplastic." And I think that kind of sums up the situation. But if you're talking about the suite of contaminants, which is really what microplastics are, they're defined as plastic particles that are between 5,000 and 1 micron in size. And that's a pretty broad range. As you get working on samples, if you're collecting samples from the field, if you're looking in biota or sediment, often you'll find that you're at the smaller end of that scale, and that the samples and the plastics that you'll spend the most time on are the ones that are 100 microns and smaller, because those take more work to classify and to analyze. There are primary and secondary microplastics. Primary are the beads that they used to put in a lot of cosmetics for exfoliation purposes. They're also still added to lots of other products, industrial sort of abrasives that are used to break up pavement. Microbeads are added to some kinds of paint. Those would all be considered primary microplastics, whereas secondary microplastics is basically everything else, or mostly everything else. So anything that's breaking down from a larger plastic, from fragments that are coming off of a plastic bottle that's been left at the beach, to fibers that are sloughing off of your fleece or coming off of your clothes when you do laundry and then washing out with your house's gray water. Those would be considered secondary microplastics. And they've, of course, been found in a wide variety of environmental compartments and products sold for human consumption. So it's not just about them being found in fish, in the ocean, in fresh water. They're also found now, it's known that they're found in honey, in beer, in drinking water. There was one paper that showed that sometimes plastic counts can be higher in bottled water than they are in tap water. That's still a bit up for debate, but it's in the air that we breathe. So they're found everywhere we look. And it means that there's a lot to consider for method development, and that a lot of time needs to be spent on developing really kind of bulletproof protocols, so to speak, when you're getting started and starting to do microplastics work in the lab. And so making sure that you don't have a lot of background contamination coming in, making sure you're wearing all cotton lab coats, that you have hair tied back, that you aren't contaminating your samples in some unintentional way, or if you are, you're accounting for that contamination. And really, and this is becoming less true, but in some ways microplastics and nanoplastics, which Dr. Stacy Harper is going to talk about in an accompanying short course, they're sort of the new pollution. And I think you can still say that, even though we've been studying them pretty intensively for about 10 years now. As you can see from this graph, the number of papers from 1980 to 2010, so that 30-year period, was pretty small, even when you looked at just basic papers on occurrence in water, occurrence in biota, and almost no papers considering the potential for effects in organisms. And then you get to the past 10 or so years, and you see that rate of publication has just really increased exponentially. But in the grand scheme of things, when you think about how long we've been studying other types of chemicals, think about endocrine disruption research, or the amount of time we've been studying pesticides and the potential effects of pesticides on organisms. 10 years is not really that long. So even though plastics have been around for longer than most of us have been alive, and we really started to produce plastics around the time of the Civil War, when tires started to be produced for bicycles in larger quantities. As we all know, the science is still relatively young, so we still have a lot to learn. And like I emphasized earlier, this is really a diverse group of contaminants. It's not just one contaminant. If you study the effect of a polystyrene particle on a fish at one size, at one concentration, one color, one shape, you're not going to be able to draw conclusions about all of the microplastics that exist. And as you can see from this chart, which is from that same paper I mentioned earlier, you have this exhaustive list of polymer types, additives that are used from plasticizers to fillers to flame retardants, types of products that they come from, which can influence how they fragment and what shape the secondary microplastics that break down from them are. You've got different sizes, from micro to nano, morphologies, and sometimes that shape, there are at least indications that the shape can influence whether a particle is toxic or not, and where it's toxic, where it ends up, where it gets stuck. Colors, and then at the end of the chart here we have ecotoxins. And an ecotoxin, or toxicant, is something that is in the water that sorbs to the plastic after the plastic has ended up in whatever environmental matrix it ends up in. So it could be in the water, it could be in the sediment, it could be in the air. There are other persistent pollutants that are in those matrices that often will preferentially sorb to the plastic because the plastic is also hydrophobic. Sort of the opinion that is becoming more common in the field is that we maybe don't need to be as concerned about those persistent pollutants that sorb to plastics as we do about the shape of the plastic, the concentration and the size, which may have more of an influence on toxicity than the very small amounts of pollutant on the plastic that leach off into an organism. And that's still a bit of a debate in the field, but additives are potentially of more concern because they're added at much higher concentrations. And so that's something that still requires some study when it comes to how much of a concern we should have for those associated pollutants that sorb to plastics once they're in the environment. But that's a whole other class. So because there are so many types and sizes and shapes, it's not possible to really designate just one water quality criterion or one threshold that applies across all plastics. We really need to think about each plastic type as a distinct type of pollutant, just like when we analyze pyrethroid pesticides for potential toxicity, we look at each pyrethroid pesticide separately. So the same thinking goes here, but there are many more possible combinations. So the physical characterization of microplastics is really an art in and of itself. It takes a lot of time. You need to think about the size, the shape, and surface morphology. For smaller particles, smaller microplastics and nanoplastics, surface charge can be very important, as well as functional groups that are added to that particle. Color can be important, depending on maybe what organism you're having that plastic interact with. Some fish are visual predators, and so color can make a difference. Material type. There are some anthropogenic particles that are technically not plastic as you would think of plastic, like a plastic beach ball or something like that. But textile fibers that are made from synthetic materials, like polyester, and tire wear, which is really at this point mostly synthetic anyway. It's a compound called styrene butadiene styrene. Those are also now kind of lumped in with the broader category of microplastics, and tire wear has really become a big issue just in the past couple of years, as we realize that many urban waterways are highly contaminated with tire wear particles from our cars. So a few different examples of microplastic morphologies, which I know my students are all pretty familiar with at this point, picking a lot of these things out of samples. You have fragments, which are kind of secondary microplastics that are breaking off of a larger piece usually. Those are pretty common in the environment. Spheres are less common in environmental samples, more common in the form of primary plastics or nurdles that are melted down to make larger plastic items in different shapes and sizes and whatnot. Pellets, as you can see here, this is a little bit larger. And of course, fibers and fiber bundles. We'll say from my short experience, and when I say short, it's been about 5 years, so about half the time we've been intensively researching microplastics as a larger research field. Fibers are far and above the most common thing we find in most environmental sample types, in fish, in water, in sediment as well. Sometimes they come in bundles, so they're kind of stuck together. This almost looks like it came out of someone's dryer or washing machine filter. Foams, so this might come polyurethane, potentially it might come from a mattress. And then films, so things like cellophane, wrappers, packaging. Packaging is one of the most common types of plastic pollution, is often found. So each of these morphologies, and you can, looking at them, it's pretty obvious that this would be true. Each of these morphologies interact differently with an organism that ingests or inhales it. The shape can dictate what the plastic ends up doing in that organism's body, what types of potential toxicity it exerts, how easy it is to excrete. So an example of this is I'm showing some work that we did a couple of years ago here, where we exposed juvenile sea bass to different shapes of microplastics. So they were exposed to a couple of different types of microspheres. One was clean or virgin. The other two had associated pollutants added to them. And then the fourth type of plastic was a fiber, it was a polyethylene fiber that didn't have anything added to it. And fish were exposed to these 4 different plastic types for 96 hours. And then we did a respirometry measurement. We used this enclosed chamber to measure rates of oxygen consumption. This is a closed system respirometer, 2 fish per replicant, normalized to body mass, did kind of all the standard adjustments that are done in respiration experiments. And what we found was that only the virgin microfibers caused the fish basically to labor more for oxygen, and so they were working harder to breathe. Our hypothesis is that those fibers were potentially embedded in the lungs. Some other research has shown the same thing, and it's possible that those fibers are harder to eliminate once they get embedded in the gill tissue. And so we didn't see the effect with the spheres. We saw a very distinct effect with the virgin microfibers. And so there are a lot of gray areas when it comes to classifying microplastics. And different groups are applying different criteria to classify whether something is a microplastic versus a nanoplastic. There's still some disagreement in the field over what the cutoff should be. Some groups are using the analytical capabilities of our current instrumentation that is used to identify polymers to type as a way to differentiate between micro and nanoplastics. We're able to analyze plastics that are 1 micron and larger using kind of standard instrumentation like FTIR and Raman, as well as pyrolysis GCMS, which I'll talk a little bit about later in the course, whereas particles that are smaller than 1 micron are a little bit more, they're more difficult to analyze. They're difficult to pull out of the environment and identify. And so most of the work done on nanoparticles, for that reason, is done with particles that are purchased commercially and then used in laboratory exposures, rather than looking for them in biota or water samples or sediment samples. But as you can see here, this is a proposed definition and categorization framework that came out in 2019 from Hartman et al. And they had a couple of different criteria. One was chemical composition, one was that it was a solid state, and then they had this third criterion for solubility to include soluble polymers. And then here they have nanoplastics as defined between 1 and less than 1,000 nanometers or 1 micron, which is the definition we use in our lab. But some groups use a definition of 100 nanometers and below being a nanoparticle. And then you have microplastics, which are 1 to 1,000 microns, which is different than NOAA's definition, which goes up to 5,000. They have mesoplastics as being 1 to 10 millimeters, and macroplastics as being 1 centimeter and larger. So this is a little bit different than some of the definitions typically used by labs in North America, but groups are gradually converging on kind of a standardized way of defining these different sizes. But there's still room for debate and room for discussion. So we're reconsidering the criteria by which we classify what is plastic pollution and what isn't, and then from that we can build a framework to assess toxicity from the ground up. So standardization is ongoing, and it's complicated, and people spend many hours and days and weeks talking about this challenge and trying to come to some agreement and consensus on how we best standardize plastics. And you can see from this list of papers, starting with Gregory and Andrade way back in 2003, so way before those publications started going up exponentially, and then all the way down here to EFSA in 2016. And some of these are European regulatory groups. You can see the chart here has nanoplastics, microplastics, mesoplastics, and macroplastics. And each group has a slightly different cutoff, which can be a little bit confusing. There are some groups that classify nanoplastics as being 1 to 100, and then there are others that classify nanoplastics as being a little bit larger, up to that 1 micron size. And then the cutoff between mesoplastics and macroplastics can also vary. But typically in North America, the less than 5 millimeters is a microplastic. More of the debate is between what where that nanoplastic cutoff is, whether it's at 1,000 or 100 nanometers. So just something to be aware of when you're going through the literature. You may see different definitions depending on where the paper was published. So 1 micron, like I was saying earlier, is the current cutoff for micro Raman spectroscopy. And so if you have a particle that is smaller than 1 micron, you're not going to be able to identify it with any currently available instrumentation, at least if it's something coming from the environment that you don't already know is in your sample. But there are methods being designed as we speak to be able to detect these smaller particles. So what about natural materials? It's really easy when you're searching through the gut contents of a fish and you're trying to figure out what's microplastic and what isn't, to mistake things that are not plastic for plastic particles or polymers. Salt and things like sand are sometimes pretty easy to differentiate because salt will break and can be crushed. It's going to dissolve in water. Sand is pretty usually clear and brown and really hard. So you can tell something is a plastic sometimes if you tap it and it gives a little bit. It's a little bit spongy almost. And sand can break. Coal dust and fly ash is easily crushed. So things that are very brittle and easily crushed are often not plastic. Not always. Sometimes you'll have something that breaks that is plastic. We know that plastic degrades gradually in UV light and with wave action over time, so it can break when you touch it, but there are kind of some general rules you can use to differentiate between a plastic debris item and something that's natural, like shell and bone, algae and plant matter, as well as microscopic organisms. So here you've got a diatom, a trichome, and a copepod, and you can, and it's a lot of very painstaking work in some cases where you're spending a lot of time over your microscope, very carefully picking through your sample to differentiate between plastic particles and natural particles. So using only visual identification can often lead to the misidentification of natural materials as plastics and vice versa. And so most groups do not just use visual identification. They also use some sort of instrumentation for analytical confirmation that those items that they designate as potentially being plastic are actually plastic. And that's really been a shift over the past few years in the field, in that studies that used to just use visual identification, used microscopy, are now not as readily accepted in terms of publication in a peer-reviewed journal, and having some sort of analytical identification, at least for a portion of your samples, is really important. I'm sorry, my cat decided to visit us for a moment. All good. So, of course, the only way you can identify your plastic is to actually get it out of your sample. And so a lot of time and thought and publications have come out about how to best extract your microplastics. And before you can extract them, you usually need to break down your sample in some way or separate it in some way so you can get to the particles that might be in there. So a lot of groups use chemical digestion. This is often in the form of using potassium hydroxide. Some groups use hydrogen peroxide. Typically, potassium hydroxide works best for biota, for tissues from organisms. It also can be used for scat, for example. Hydrogen peroxide, I've seen that used for water samples. And so it's sometimes a matter of trial and error and figuring out what works best for your particular sample type. For biota samples that have a lot of lipid in them, sometimes it helps to add a detergent, and that detergent helps to kind of break up the lipids, just like it would if you were washing dishes. You can think of it that way. It breaks up the lipids so it's easier to to get them into solution and to digest that sample. And excuse me, there are lots of different applications. There are limitations to all of these different approaches. And another tactic that is used on some samples, particularly those that have a lot of sediment in them or a lot of natural material like shells, density separation works well on those samples. So using a salt, good old sodium chloride works pretty well. Some people use calcium chloride or zinc chloride, which is a bit more expensive, but can sometimes be more effective. And that helps to float the polymers to the top of your sample, since those tend to be less dense than natural items that would be more likely to sink out, like shell and bone, pieces of exoskeleton if it's an animal that eats crustaceans. So those are kind of the two main first goes at your sample. You want to chemically digest it in some way, and then if necessary, do a density separation so you're able to get at the fraction of the sample that is most likely to contain your polymers, if there are any there. And then after that, sieving is really important, and filtration. And so depending on the size of the particles in your sample, using a stainless steel sieve to kind of filter out any other excess sediment or roughage that's in that sample that isn't plastic. And then a lot of groups will do kind of a staged filtration. So filter through, say, a 50 micron filter, a 20 micron filter, and a 10 or 5 micron filter, depending on what size you're going down to size fractionate your sample. And so you're able to look at plastics of different sizes when you're doing your microscopy and eventually your analytical identification. So many labs use a combination of these techniques. Usually you have sieves of a couple of sizes, and like I was saying, sometimes material is separated by size. Generally, vacuum filtration should account for particles 1 micron and up. Sometimes in my group, we only go down to 20 microns just because of the analytical equipment that we have. We're not able to get down to those very small size fractions because we don't have a micro Raman. So you want to tailor your approach to your sample type, and that can vary quite a bit. And it's common that the method development stage of a microplastic, project is going to be a considerable chunk of time in the beginning. But once you get it going, and once you've sort of figured out what the best approach is for your particular sample type, then it goes a bit faster. But that method development can take some time. So, like I was saying earlier, QA/QC is super important when it comes to microplastic work. And so I'm going to talk a bit about some of the best management practices for robust microplastic method development. And some of what I'll talk about is coming from a special issue that a working group I was part of put out in 2020. So it came out in about summer, fall of 2020 last year. So we know, we've talked about this already in this course, plastics are everywhere. The smaller the microplastic that you're aiming to pull from a sample, the greater level of what I would call pre-experiment planning is needed. You're going to need a more complex experimental design. You're going to have a greater need for QA/QC throughout every single step of your experiment or your extraction. And this is a picture. My colleague Jeremy Kunkel sent this, or it was part of a presentation he gave. I think this is his son, but just showing that microplastics are everywhere. They're in the air around us. They're coming off of our clothes. They're coming off of the electronics in our home or in our lab. We're bringing them in on our shoes. And so you need to think about everything kind of being contaminated when you're working on samples and looking for microplastics. So this is basically Oh, this was from last year when we taught this in person, but what we did last year was we placed a petri dish with wetted filter paper in the back of the room where we were giving the short course last year. And at the end of the course, people looked at it under a microscope that we had brought in. And of course, lo and behold, there were mostly fibers in there, probably from people's clothing. But we definitely had some background contamination in the room that the conference was being held in. So a colleague of mine has called this the pig pen effect. And if any of you have watched Snoopy cartoons or Peanuts cartoons growing up, you know who Pigpen is. He's the character who, as he walks around the neighborhood, there's dust and dirt and all sorts of debris sort of sloughing off his clothes. Well, that's happening with us too, but to, of course, a less visible and much smaller extent. But considerations for sampling in QA/QC measures associated with sample processing ideally ought to be determined at the very onset of a study. And that's why I was saying earlier that method development phase, it's okay if it's a little bit longer than maybe you anticipate, because if you spend the time to develop those considerations, you're going to have less of a chance of getting midway through processing your samples and realize there's something that you hadn't accounted for. There's a source of contamination that you forgot to measure, for example. And so the goal of this recent working group was kind of to provide a checklist and how-to guide, because there wasn't one out there. And so what you're seeing here is from one of the papers that was published in that special issue. This was in the Journal of Analytical Spectroscopy in 2020. And so you can see for sample collection, you want to be thinking about everything from gear type to season and weather, because microplastics can differ depending on whether you're sampling in the dry season versus the rainy season, or whether you're sampling in warm weather when animals tend to be feeding more because it's breeding season, or whether you're sampling in cold weather when they're maybe a little bit less active and consuming less food, and so they're being exposed to fewer microplastics in their diet. So lots of considerations with sampling, as well as lab processing. So how are you going to store your samples? How are you going to filter them, homogenize them and digest them, extract them? Will you be subsampling? How many samples do you need to process? Make sure you have enough replication. If possible, try to create a situation where you can composite samples if you're collecting them in a small area where you can have that level of replication. Thinking about secondary contamination is something I think about now pretty much every day. So you want your equipment to be non-plastic when possible. Clean it, pre-filter your solutions, modify your lab space, have sticky mats at the door so you're catching some of that debris that comes in from people's shoes. You want to make sure you have clean working surfaces. For your blanks, you want to have blanks in the field and in the lab, and have those blanks out whenever you're processing a sample so you can account for what might be in the air or elsewhere in your space. For sample recovery, and this is a little bit less common still, but there is discussion and efforts being made to generate standard reference materials, just like you have for other classes of pollutants like perfluorinated chemicals, for example, or pesticides. People are starting to think about doing matrix spikes. For example, I'm part of another working group now that has sent out spiked samples to a number of different labs, and they're doing an interlaboratory comparison to see how much difference there is in detection and accuracy between 20 or 30 different lab groups when they process the same samples. Some groups have positive and negative controls. This is really important, of course, if you're doing exposures in the lab. You always want to have a negative control. If possible, have a positive control too. Some groups are using natural particles that are the same size as the polymers to kind of have a positive, well, this is just a natural particle, but it's the same size and same amount as the plastic. Do I see the same kind of response? and that's becoming more common. And then data analysis and reporting. You want to make sure that you are, even if you aren't correcting for your blanks or subtracting the plastics that are in your blanks, and this is still an area of discussion, you want to at least report the number of plastic particles that you have in your filters that you leave out to see how much contamination is in the air, for example. Some groups are starting to think about limit of detection and limit of quantification. That, of course, gets complicated when you're working with samples from the field, because there could be 600 different types of particles in your samples. You're not necessarily going to be able to come up with a limit of detection for each one of those, but that's something that is being discussed, and people are thinking about how to approach that situation. And so this is sort of the list of things to consider when you're thinking about designing a microplastics experiment, or you're about to embark on a microplastics project. From the outside, it probably seems pretty simple. Oh, I'll just grab a few fish and count how many plastics are in there, done and done. And it's to do it right, it's a little bit complicated, and it takes a little bit of time and consideration. So, thinking about field and lab prep, something that people often don't realize, and that we didn't realize at first too, is that DI water is very typically contaminated with fibers. Fibers are everywhere. They get in everything. An experience we had in our building was that they came in, this was a year or two ago, and vacuumed all the carpets in people's offices. And even though the lab was tiled and didn't have a carpet, because they were vacuuming the carpets in offices in the same building, those fibers from the carpets then got caught up in the HVAC system and ended up, in our background controls, in our blanks. And so just things like that you wouldn't normally think of can interfere with your sampling. And so always accounting for that background is really important. So DI water should be tested for fibers. Even RO water can sometimes be contaminated. That's reverse osmosis water. Field gear, it's important to replace materials that might shed. You want to minimize the use of plastic where you can. I think I said that earlier. And know what materials you're using. Wash your gear thoroughly. Rinse it off. And rinse it off with water that is less likely to be contaminated with particles. Avoiding synthetic clothing is really important. Fleece is like a microplastic researcher's worst enemy. As much as fleece is great, I live in the Pacific Northwest. Fleece is the best thing to keep you warm on those cold, damp days, which there are a lot of in Oregon, but it's really pretty bad for microplastics research. So wearing, instead of wearing fleece, wear a coat. It could be plastic, but make it, something that isn't going to shed. So, a raincoat or something like that, as opposed to a fleece. But accounting for that background contamination wherever you can is really important. The nice thing about having something like an FTIR or micro ramen in the lab is that if you suspect that something you're using, maybe you can't avoid all plastic, something you're using is maybe contributing contamination to your samples, you can identify that polymer type using FTIR or ramen, and then if you find a particle in your sample from that polymer type, don't count that as being from the ocean or from the river or wherever your sample came from, or subtract that from your background. So sampling design should really be informed by the source or origin of the microplastic debris, the pathway by which it traveled to a particular location or facility, as well as the matrix, be that air, water, biota, or sediment of interest. And this is just a diagram that we had in one of those publications that came out with a special issue, just thinking about all the different sources, the different pathways, which can range from wastewater to biosolids to wind to runoff, all of the different matrices. And then thinking about your question or approach, what is the scale at which you want to measure microplastic contamination? What about particle size? What sample preparation is going to be required? And what analytical method are you going to need to identify whether those particles that you've pulled out of your sample are plastic? And that's something that's really important to think of early on, because the analysis is often what ends up being most expensive. Most studies are nowadays are relying on some sort of spectroscopy or GCMS to identify at least a portion of their samples. Generally, 10% is kind of the bare minimum of your samples that you want to identify chemically. And you might try to budget to purchase an instrument for your lab, which is not cheap, but possible. That's what we ended up doing at OSU. Or there are a number of different labs, including ours, that will process samples for external studies for a fee. But just something important to keep in mind, because it's something you would need to budget for, to plan for, and you want to make sure you can analyze an adequate portion of your samples in order for those data to be publishable. So research approaches are really best rooted in standardized techniques that simultaneously address scale, a targeted range of particle sizes, and then again, like I was saying, forward planning for sample prep, ID, as well as analysis. So there are roughly 6 categories of sample types when it comes to microplastics. Drinking water. Typically, drinking water, you're going to find really small microparticles. Some studies show that there's more in bottled water, but it's also present in tap water. I make a note that this will be subject to regulation soon in California. When I say soon, I mean later this year, but that would be a whole other presentation too. Wastewater. Tons of regulatory implications, lots of cellulose from toilet paper. And then this type of sample is also susceptible to contamination from plant workers, from tubing in the plant, lots of other things. Biosolids and sludge are becoming a big topic of discussion, because it turns out that most of the plastics in wastewater end up being deposited in the biosolids. They sink out into the biosolids rather than mostly ending up in the effluent. Those biosolids are applied to agricultural fields, and then they end up being reintroduced into the environment, particularly during storms and stormwater runoff. So that's a big area of terrestrial investigation now. And surface water. There's a big challenge between the volume needed versus the ability to detect particles smaller than a net mesh size. And so some recommendations for surface water are now to use things like auto samplers, instead of nets, because a lot of the nets, plankton nets used in early microplastic studies had a mesh size of about 300 or so microns, meaning that anything smaller than that went through the net, and you weren't accounting for those smaller particle sizes in your study. Air, of course, is a big, relatively new area of investigation. We're finding microplastic particles pretty much everywhere we look, from the Pyrenees to the Rockies to, of course, there was a famous study that was done in Paris that found pretty high concentrations in it. Indoor air may be more, or I shouldn't say may, probably is more contaminated than outdoor air. And then sediment is a sink for plastics denser than seawater. We know that there are estimated to be about twice as many microplastics on the floor of the ocean than there are in the surface, as well as those plastics with biofilms, for example. That's going to change their density, and they may be more likely to sink. And then, of course, biota, maybe one of the bigger areas researched now. It's important to collect enough animals per site, account for home range size. Is it an animal that tends to swim around large distances? So you need to account for migration and overlap with animals from other sites. seasonality, like I was mentioning earlier, as well as sampling across depths where possible. And then there's a lot of work now on scat. For example, a student in my lab does research on otter scat and looks for microplastics in scat. And so another sample type to think about, and that is complicated because it needs to be fresh in order for it not to have been contaminated by microplastics from the air, now that we know that there's a sizable amount of microplastics in air too. So lots of different categories. And I really like this diagram, and I like to talk about this paper a little bit because it is really kind of daunting to think about sampling in some of these situations. And regulation at wastewater treatment plants may also be something that comes to pass in the near future. And wastewater treatment plants may be required to monitor both their effluent and biosolids to get an idea of how much microplastic contamination is exiting their plant following treatment. And so this is from a paper that was published in 2016, but then we included this figure in the paper that came out in 2020, not in review. It's actually published in the Journal of Applied Spectroscopy. And you can see when you're thinking about sampling at a wastewater treatment plant, and primary, secondary, and tertiary process points are noted above. And then those green stars are recommended sampling locations. And what the arrows describe is the direction of flow of the waste that's being treated. And you can see that removal efficiencies from wastewater into sludge are about 88 to 97%. So like I was saying earlier, you tend to find a lot more microplastic in the biosolids than you do in the effluent, in the aqueous part of the treated component that comes out of that plant. And then in secondary or tertiary treated effluent that is relatively clean, just to put this in perspective, you really need a minimum sample volume of 20 to 30 liters to provide counts above minimum detection limits. And so it's really quite an undertaking to sample at a wastewater treatment plant to really get a solid idea of how many microplastics are present in effluent, how many microplastics are present in sludge. It really needs to be a multi-dimensional sort of sampling design with consideration of all these different processing points in that plant. And so you can see how much work this will be for plants once they're potentially required to report how many plastics are in their effluent. And it's a good goal to have. It's good that we're thinking about this as a source of plastic to the environment, but it's a lot of work to put those types of monitoring programs into place. So, and then of course, you also need to consider false positives from uniform shedding. I know that talking to collaborators at the Seattle Aquarium, they ended up changing their uniforms because they found that their uniforms were shedding a lot of fibers into water. And the reason they found this out is that they started studying microplastics in otter scat in the otters at the Seattle Aquarium. So just things that you would never think about unless you were trying to measure how many microplastics were in something. So what about field sampling? And this looks like a fun project they're doing in this picture. So they're just kind of common sense things, cleaning and rinsing your sampling equipment between samples, having field blanks. So for water sampling, have an open sample container with DI water or Millicue or RO, whatever your clean water source of choice is. For sediment, you can use pre-combusted sand. For organisms, it's not really possible to have a blank fish or a blank crab, whatever it is you're collecting, but you can at least collect background contamination. And you want to have at least one field blank per day, or 1 per 10 samples is kind of a general rough rule. For equipment blanks, it's important to run clean water through equipment as if you were collecting a sample. This is something that is also often referred to as a procedural blank. Sediment, excuse me, you want to clean equipment as you would between samples, but then do a second rinse with DI or MQ or RO water to check for any carryover, anything that got left behind. And having at least one equipment blank per day or 1 per 10 samples is important. It's not always realistic to have 1 per day. And triplicates if possible. So for lab processing, really important to be wearing personal protective equipment that is microplastic compatible. So you want to have a buttoned up fitted coat, but it should be cotton. And surprisingly, it is not easy to always find 100% cotton lab coats. You can purchase them through VWR or Fisher, but often the companies that larger universities use to provide lab coats to labs that rent them from the service provider. What we found at OSU was that all of those coats are made from polyester, and that there wasn't an option to rent cotton lab coats from the university. And so we ended up having to buy them. And now about once every couple of months, everyone puts their lab coats in a bin. It gets sent home to me. We run a blank load, basically just with water through our washing machine, wash the coats, and then ship them back to the lab because there's not a service that will provide cotton lab coats. They do provide cotton jumpsuits, but they're only designed for men. And so anyway, sometimes there are little workarounds you have to come up with in order to deal with some of the requirements necessary for working with microplastics. But it's possible to make do. Try to make sure your pants and your shirt are cotton as much as possible. Closed-toed shoes, we wear gloves. Something important to note about gloves, though, is that it is a good idea to run your gloves, if they're nitrile, under an FTIR to get the spectra. So you make sure that you're not pulling little tiny pieces of gloves out of any of your samples, because sometimes that can happen. But, just have your lab equipment blanks, your lab sorting blanks, and again, a minimum of 1 blank per 10 samples or per processing event if possible is great. More blanks is better, but not always realistic, as you don't want to spend all of your time processing blanks. I'm going to get to your samples too, but important to keep track of what might be in your background and your surroundings in the lab. So what about sample processing and sorting? Important things here too. I'm thinking about method validation, testing your sorting or vacuum recovery. Some groups will spike particles into a sample, count how many they put in, and then run it through those digestion and vacuum filtration steps, and then see how many you pull out at the end. Is it an 80% recovery, a 90% recovery? That's pretty good. If it's 30%, that's probably not good, and you might need to redesign your approach. Training new researchers, just like with any other lab procedure, you need to make sure people are taught the basics, walk through methods, have them try it, and have them try it again. And that's just kind of, common sense and something that most people do when they're starting any new experiment. I am going to show you a video that my grad students made in the lab about a week ago, just kind of walking you through a sample. This is how I process one in this case otter scat sample that was collected from an estuary in Alaska. So I'll go ahead and play this video. It's only a couple minutes long. And this is my graduate student, Jennifer Van Brocklin. She's a master's student in the Fisheries and Wildlife program at OSU.
So here I'm working inside one of our laminar flow hoods with a HEPA filter that helps prevent much of the plastic contamination in the air from reaching the samples. I am processing spraints or scats that were collected from wild sea otters in Alaska. I have dried the spraints, digested organic material with potassium hydroxide, and performed a density separation to try and remove the potential plastics from the shells and sediment. Here I am sieving the remaining sample through a 1 millimeter sieve to separate the larger shell pieces from the smaller sediment so I can look for any plastics that might still be in the sample. After this step, I will vacuum filter all of the liquids from this sample through a 20-micron filter and analyze this filter under a microscope, picking off any potential plastics I find. These particles will be photographed, measured, and categorized. Then a subset will be analyzed with the micro FTIR to confirm their identity.
Right, and you can see that that is a process that she goes through with every sample that she collects or that she's had sent from Alaska. And so it's a lot of very detailed work, which, you know, many protocols for many different types of lab work are. But it takes a lot of focus and attention to detail, not surprisingly.
So
what about quality control? Well, you know, most groups, most academic labs, as well as industry labs and government labs that are doing microplastics work, really try to implement GLP or good laboratory practices to minimize contamination on surface and from clothing. And these are things that I've already mentioned earlier in the presentation, but you want to avoid plastic where possible, include FTIR spectra of gear or plastic containers or your nitrile gloves, clean glassware meticulously. We heat our glassware in a furnace. So we heat it to 450 degrees Celsius before we use it again. So that's part of the cleaning process. Filter your liquids and use HEPA filters or HVAC systems when possible to remove lab air contamination. So this is a laminar flow hood that's pictured in the background here, and this is my student Catherine Lasden processing a rockfish sample. We also even have a HEPA filter in our regular chemical fume hood where less work is done, and that's really only used for vacuum filtration where things are covered, but it's a way to have another kind of clean space to work in that has reduction of potential air contamination. Use background checks and controls and procedural blanks in the lab and in the field. Usually just a piece of wetted filter paper will do. And then, like we've talked about before, using procedural blanks is really important, and you can use these to determine the limit of detection and quantification. Some labs are using an average of blanks to determine LOD and LOQ, but it's, like I was saying earlier, it's more difficult to do for plastics compared to soluble pollutants because you're dealing with so many different potential types of plastic in your blanks as well as in your wild samples. It can sometimes be easier if your blanks end up being the same plastic type every time when you get something in there. So because our building has blue carpets, there are often blue fibers in our blanks because it's just part of the, it's getting circulated in the HVAC system. And so it's easy to say, oh yes, that's the typical blue fiber we see in our blanks. And so we're not gonna count that if we see it in one of our samples. But having an LOD and an LOQ for each plastic type can get a little bit tricky. But you know, that being said, one study that was published in 2017 reported dry deposition rates of 1,600 to 11,000 particles per meter squared per day for indoor air. It's going to vary a lot depending on the building you're in. I don't think ours is quite that bad, but just to get the point across that having some sort of filtration or clean space to work in is really important. So what do we do with the blanks? And this is one of the biggest areas of concern because we have shapes, we have different colors, we have different polymers that are identified using spectroscopy or pyrolysis GCMS if you have that available. Blank correction, you can subtract based on known characteristics like blue fibers or, black fragments. And you can have specific blanks associated with specific samples. It works better when you have greater than one blank per 20 samples. And I think there was a rule of 1 per 10 from earlier. You can pull the materials from all blanks and then perform correction based on those combined blanks or your average background contamination, which is another kind of conservative approach that people use. So if you've been processing samples for a year, grab one blank per week and look at what your average contamination is and what it's comprised of. So some groups are using spiked recovery. This still is a bit uncommon in the microplastics field. And this can include creating representative standard reference materials that contain microplastic particles to be used to spike into representative matrices. And that's something that's starting to be done by some groups. Like I was saying, I'm involved in a study where a group has sent samples out to different labs, and we're comparing recovery and accuracy and identification between those labs. But it's not quite something that individual labs are doing yet. Spiked reference materials could, in theory, be carried through the extraction and preparation process, which is what we did for this interlaboratory comparison. And then you want to show, ideally, a recovery rate of 80% or higher. For some microplastics, this is pretty difficult, especially for fibers, which tend to do sometimes whatever they want to do. They don't behave the same as a fragment or a sphere because they can change they can move around and get stuck in a sieve or go through a sieve when they aren't supposed to. And so that's still something that is up for discussion when it comes to how do we best handle things like fibers. But the general agreement is that all of these recovery protocols were designed for soluble analytes, kind of your traditional analytical chemistry. And microplastics are a different animal, so to speak. It's a three-dimensional object. They're different shapes. It's not going to behave the same way DDT or PCBs or organophosphates behave because they don't dissolve in water. And the Southern California Coastal Water Research Project is the one running this large interlaboratory comparison using pre-spiked samples, and that's been happening since late last year and will go through the rest of 2021. So some general lab practices, and again, some of this is a little bit repetitive, but processing in a separate clean room or hood when you can. Avoid lots of opening and closing doors. You can install sheet curtains, have HEPA filters, regularly clean the room. Like I was saying earlier, your clothing should be made of natural fibers. Some groups have even dyed their coats bright pink or bright orange, some sort of color that you don't typically detect in a sample. So when you do see a bright pink or a bright orange fiber, it came from your cotton lab coat and not from the ocean or from a river or whatever it is you're sampling. Keep samples closed and equipment covered, unless it's necessary to open. Just kind of common sense things, try to think about all the different scenarios or ways your sample could become contaminated, which can be a bit stressful, but also you will thank yourself when you get 6 months into processing your samples and realize that you have thought of most of those potential caveats and challenges when it comes to background contamination. So just kind of a general, getting from here to there. When you're collecting your samples, you want to make sure you're using clean containers, keeping things covered. Sample preparation, have your lab blanks, reduction of plastics in everything that you do. Sample analysis, make sure you have access to instrumentation to confirm the material type. It's a really important step, and again, it's hard now to get your data published unless you have a portion of those data analytically confirmed. And then for data reporting, I would say LOD and LOQ are not standard protocol yet, but they may be in the future. But just, be honest about the plastics that were in your blanks. Report what your background contamination was. So lastly, I'm going to talk a bit about different analytical methods. And you've heard me refer to FTIR and micro-Raman and pyrolysis GCMS a bit throughout the presentation. But microplastics methods are diverse, as you've learned so far throughout this course. And methods can really vary with particle size, with the types of plastics or anthropogenic particles targeted, and here they may be referring to something like tire wear particles, which are now just lumped in as another type of microplastic. Shape, what environmental media did it come from, what is the cost, what is the instrument availability, and sometimes it comes down to those things: how much does it cost, and does my collaborator have an FTIR or a micro-Raman? I'm going to use the one that is most readily available. And material identification, both physical and chemical, is really important. And you can see this graph is a little bit backwards, but if you look at Web of Science, starting from 2010 over here and going to 2019, you can see that the number of publications on microplastic methods is also increasing just as quickly as the publications on microplastics occurrence and effects. So physical characterization. Stereomicroscopy is very important. And you saw that in my lab, the stereo microscope is set up. So you basically have the microscope under the hood, but then you can visualize what's under the microscope outside of the hood. And so you don't have someone sticking their whole body into the hood and potentially contaminating their sample. It sets up kind of a cleaner situation. But stereomicroscopy is used, obviously, for the visual identification of plastics. There are guides that can be followed to ensure comparability and that you're doing this in a consistent way. We have a guide printed out and hung on the hood, and so you can cross-reference it as you go through samples. Is this a fiber? Is it a fiber bundle? Is it a fragment? Is it a film? And so have a representative photo of each of those plastic types next to where you do your microscopy work. And at least once per day, you're going to find something that doesn't look like any of the examples on the sheet you printed out. And so just, fair warning that is not unusual, but you do your best. Limitations with stereomicroscopy are that visual ID error is relatively high, and it really should not stand alone as an identification technique, especially when you're getting into plastics that are smaller than a millimeter in size. So considerations here are size limitations of your scope, and also consideration of color blindness and color perception varying between different personnel. And it can also vary with lighting and with different types of scopes. And I don't know if anyone remembers a few years ago the debate over this image of this dress that was posted online. I think this might have been as long as 5 or 6 years ago, but some people saw the dress as being gold and some people saw the dress as being blue. And the same thing can happen with your microplastic particles very easily. It's one person's red as another person's orange. So thinking about that variability is really important. Fluorescence microscopy is often used to increase microplastics detection in environmental media. It can be more accurate. It can save time. The challenges here are, even for dyes like Nile red, which are widely used for microplastic identification, they tend to dye some plastics more readily than others. So one plastic type may appear to be a much darker red than another. They can also stain lipids, and so there's some pre-processing steps involved to remove lipids from your sample. It doesn't provide a chemical ID. It just tells you that it's maybe plastic. And those fluorescent dyes can migrate to tissues and can also cause, they can also photo bleach, and it requires a fluorescent microscope. So there are benefits to using this approach, but also some drawbacks. And it's not considered a way to replace the use of spectroscopy or GCMS to chemically identify at least a portion of your samples. For chemical characterization, FTIR, which is Fourier Transform Infrared Spectroscopy, and it's okay to just say FTIR, this is the measurement of infrared radiation that is passed through or is reflected from a test material. And the detector produces a spectrum of peaks and valleys, and that spectra is going to be unique to each molecular structure. And so our lab uses FTIR, and I have a short video from Emily Peterson, who is our lab technician who runs the majority of our samples. And she's running samples that are usually 50 microns and larger. We have been able to get down to about 20. Getting below that is a little bit more difficult. So this is a micro FTIR, which, as you can see, has a microscope attached to it. There's also, I guess what you would call a regular FTIR, which was the first type of FTIR available before micro FTIRs came on the market. And those instruments can only analyze particles that are about 0.5 millimeter in diameter or length and up. And so the micro FTIR has a somewhat higher resolution. And if you're looking for a lab to process samples, having a lab with a micro FTIR is beneficial, because a lot of the particles you're going to find in environmental samples are going to be smaller than 500 microns, and it can be challenging to try to identify them with an FTIR that doesn't have a microscope associated with it. So I'm going to show you this short video where Emily and Jennifer will walk you through a sample, the identification of one sample.
Here, Emily is using our micro FTIR machine to confirm the identities of suspected plastic particles that have been collected from a variety of different sample types. After placing the particle, which in this case is a fiber from a zooplankton sample, on the microscope, she sets up the machine to take scans of the particle. These scans will produce a spectrum, which acts like a kind of fingerprint for different types of materials. This spectrum can then be matched to a database of spectra for other known materials to determine whether the particle is likely plastic or not, and if it is, what type of plastic it is. It looks like this fiber is made out of polypropylene. Thanks, Emily.
All right, so that was Emily running that sample on the FTIR, and Jennifer was narrating for her. So we do a lot of that on a daily basis in the lab, and FTIR is a great approach. It's not perfect. There are limitations. If you're using the ATR of that FTIR, which is basically like a little probe that you stick on top of your sample, you can only measure within a few microns of the surface of a material. And so if you have a particle that is made up of different plastic types, it's layered in some way, you're only going to get the surface. And then it's only for use with particles and fibers down to about 10 microns. With that FTIR microscope, in our experience, that limitation is more like 20 to 30 microns. It can be very difficult to physically, just the limitation of physically picking up something that small and placing it on the slide is pretty challenging. There are some groups that have automated FTIRs, which allow you to take your entire filter and sit it under the under the ATR or under if you're using reflectance or transmission, and you can analyze all of those particles, basically using different algorithms for identifying what they are. That is something that I don't have a lot of experience with, but there are a lot of publications about automated FTIR, particularly from Sebastian Primke's group out of Germany, and he also had a paper in that same special issue that I've referred to a couple of times. And so for FTIR and micro FTIR, for individual micro FTIR, the analysis of small debris can be time consuming. It's pretty accurate when you're picking up those pieces and analyzing them individually and using baseline correction and smoothing software to really get at which, sometimes you'll get a list of several different polymer types, but you want to go with the one that has the highest match. A scanning micro FTIR, which is also referred to as a focal plane array, is automated, but it can take several hours to scan an entire sample or longer. And it generates a map of data pixels. So then the time dedicated in that case is not to the picking and sorting part, but to the data processing on the back end, which takes a bit longer. So there are upsides and downsides to both approaches. Raman spectroscopy is also a really popular method that is used. This is an optical instrument that measures light intensity relative to something called a Raman shift. And so light from a laser illuminates the sample and interacts with the functional groups, causing wavelength shifts accordingly, and creates a fingerprint spectra. So it's creating a spectra just like the FTIR is creating a spectra, but using a slightly different mode. It's a way to confirm that extracted particles are anthropogenic, really important for QA/QC, like we've talked about. It identifies material type, and it often also identifies dyes or chemical additives. And the limitations are that even more so than FTIR, it's only determining the surface coating of a particle. So for fibers that are dyed or have a coating of some sort, it can be difficult to identify what it is underneath. It doesn't look at that polymeric material necessarily. Requires costly instrumentation personnel time and a spectral library. That's not really much different than FTIR though. And the lower size limit for Raman, it says 10 microns here. You can actually get down to 1 micron with Raman spectroscopy if you have a highly trained technician and a very, very clean lab as well. So what sort of parameters should you use when you're doing spectroscopy? Those can change based on the type and color of the particle being analyzed. I know for us when we're doing FTIR, we sometimes will use reflectance mode first, where it's basically getting the FTIR to act like a Raman spectrometer, where it's just looking at what's on the surface. And then after that, we put it under the ATR, which applies pressure to the sample. And so if the sample breaks or gets stuck to the ATR and we can't recover it, we still have a spectra from the reflectance. And so lots of different things to consider. A lot of it is trial and error. For example, fibers will sometimes burn depending on the material and the laser used. And so you may just have to fiddle with things, and sometimes it helps to use some practice samples before you get started on the real samples. When we first got our FTIR at OSU, I was bringing in fibers from our dryer for Emily and other students to practice on before they started processing, our valuable fibers and particles picked out of rockfish guts and otter scat. And so that's a method. I know that we were producing plenty of fibers from our house laundry, and so there was a never-ending supply of practice items for them to try out. So long story short, practice before you try it on your valuable samples. A last method I'm going to talk about that is used for chemical characterization is pyrolysis GCMS. This can also be used to characterize particles. It's also really good at identifying additives and sorbed contaminants, and it's quantitative. And the difference between this approach and FTIR or Raman is that with FTIR or Raman, you're not really doing anything to the sample before you identify it. You're sticking it on your slide and you're pointing the laser at it, or you're putting the ATR on it and identifying the polymer. Here, you're putting your sample in a vial and you're actually combusting it. And the GCMS is measuring the vapors. It's measuring what's vaporized from that sample. And so it's giving you a mix of different spectra. It could be polymer type, and again, it could be additives if they're at high concentrations, as well as sorbed contaminants if it's something that's been in a polluted environment. Limitations with every one of these methods are that they're time consuming and they can be expensive. And it's one sample at a time, which doesn't differ a whole lot from FTIR. One big benefit of pyrolysis GCMS is that it's much better at identifying really complex microplastic types like tire wear particles. Tire wear particles are not easily identified with FTIR or Raman, but pyrolysis GCMS, because it generates all those different spectra from this complex material, you can more easily identify something like a tire wear particle in your sample. But there are relatively fewer labs that use this approach, I think just because it requires maybe more training, expertise, and is a little bit more expensive. So some particle subsampling strategies, because you'll get to the end of your study and you'll have 1,000 potential plastic particles and you're like, how on earth could I possibly analyze every single one of those? And it's not usually possible to do them all. And so we subsample because spectral analysis takes time. It takes money. It takes instruments being available. Sometimes I tell my students that I wish we had 3 FTIRs so they wouldn't have to wait for one person to finish processing their samples while the other one needs to get moving on theirs. But a goal of subsampling is to gain a representative view of the population, of the greater population of your plastic particles or whatever they are, while reducing the required analytical effort. A general rule of thumb is to aim to analyze 10% of the particles in each color and morphology category. Do the best you can, is the other rule of thumb. If you can do 25%, that's great. If you can only do 8%, you can probably argue that you've done analytical ID on some of your samples, and it really depends again on, sometimes the size of your grant, the availability of the instrument, and how much it costs to provide supplies and to cover personnel time. And this is the Rockman Labs subsampling strategy, which I like. So what about reporting? Microplastics, you want to catch them all. That's not always going to be possible, but you want to report your protocols, your approach, how you accounted for background contamination, and how you analytically identified those samples. And so for sample collection, you need to report site location, device dimensions, date and time, sample size, all things that you would report basically from any field study. The additional consideration here is that you're also accounting for potential background contamination. For sample preparation, you want to make sure you describe all the steps from the initial isolation all the way down to how you filtered your sample, how you digested it, whether you used density separation. And so I like this flow chart because it really walks you through the entire process from fillet all the way down to this 1 micron filter over here. For microplastics analysis, make sure you discuss how you physically characterized your samples and also how you chemically characterized them, and if you subsampled, which you probably did, what was your rationale for the subsampling? What percentage of particles did you did you analyze from each site or each organism, whatever your sort of your I'm losing the ability to think of the word here. Sort of however you split up and designed your study, your level of replication, perhaps. So if you are running an exposure to known plastic types, and this course focuses a bit less on that aspect and a bit more on the environmental sampling side, but you want to report the age of your plastic, the polymer type. Did you make it up yourself? It's usually useful to have a scanning electron microscope image so you can show the surface area characteristics. You want to report concentration and mass if possible and relate that back to talk about how did you come up with that concentration? Is it based on an environmentally sampled amount, or is it just based on person X exposed animals to this concentration, and I wanted to see if I saw the same effect. Explain your rationale. Animal husbandry, of course, is really important too. What's your IACUC protocol if you worked with vertebrates? And there's been a lot of discussion just over the past year as scientists start to try to assess the risk of microplastic exposure. And there have been a lot of papers that have come out over the past year or two about approaches to assess the risk of microplastics. How much is too much? Are we already at the point that certain areas are polluted with enough microplastic that the concentration is high enough that it's causing some sort of adverse response in fish or crustaceans or zooplankton or what have you? And a lot of the limitation here comes from the fact that not all studies use the same assay design or use the same level of quality assurance and quality control. And so making sure that you think about every step of that process, just like we've talked about sampling in the field or processing your samples in the lab, you should be able to discuss how you characterize those particles, your experimental design, how did you prevent background contamination in your beakers. For example, when my students expose animals to micro and nanoplastics in the lab, they use an enclosed kind of chamber to do those exposures in small beakers, so those beakers are not exposed to plastics in the air that are coming from other sources. And all of this is really to make sure that it's as ecologically relevant as possible and controlled, and B, that when you have all of these various controls and considerations accounted for, it's more likely that your study will be acceptable for use in risk assessment. Because when you're assessing the risk of a particular contaminant type to organisms, you need to be able to compare those studies pretty closely and be comparing apples to apples, so to speak, when you're generating those safety criteria. And so that's, again, the quality or the risk assessment process is, I would say, in its early stages, particularly considering how young the field of microplastics research is. But it's a question that a lot of people want to answer because industries want to know if they need to modify their products. Wastewater treatment plants and drinking water plants need to know if it's necessary to regulate microplastics, if the amount that they're seeing in their plant is too high. And so there's definitely a sense of urgency to get to that stage where we can assess the potential risk of microplastics. So the more studies that are able to get this adequate QA/QC score, the better. So this de Ruyter paper that came out in Environmental Science and Technology in 2020 is definitely one to read before you design your next experiment. So reporting QA/QC practices, QA/QC is often a standalone section in a manuscript where you're describing all of the different components that we talked about today, and that's pretty normal. And this is normal in the field of analytical chemistry. For the microplastics field, I think you have a lot of people like me who are coming at it from more of either the biologist perspective or the ecologist perspective. And so we're sort of learning some of these QA/QC practices secondhand. And so it's important to remind everyone that this can be really a separate and sometimes a lengthy section of your manuscript, just to make sure you're doing everything up to standard. So to summarize, and I realize that was a long presentation, a lot of information. It's all recorded, so if you need to rewind and rewatch something, you're welcome to do that. But much of what is detected in environmental samples is what I call microplastics, so far smaller than 1 millimeter. And nano remains too small to detect in environmental samples. Hopefully that won't be the case for too much longer. Careful selection of sampling regimes will allow for a greater range of microplastics to be accounted for. Better standardization across laboratories allows for much needed meta-analyses and more reproducible data, and that's important for studies from the environment, and it's also really important for toxicology experiments. And ongoing work on how to best approach LOQ and spiked recovery for microplastics, as well as the percent analysis required, is, it's still something that I think is going to be discussed over the next few years. And we really need some more interlaboratory comparison studies, I think, before we can really come to some sort of final agreement on whether we need to use spiked recovery, whether LOQs and LODs should be implemented for microplastic studies. We need a lot more data for terrestrial organisms and sediments, biosolids. Plastic pollution on land may be greater than that in marine and freshwater ecosystems, as well as in the air. This field started with a focus on the oceans, on marine microplastics, but really it's shifting to include analyses of matrices from every space, because we know that micro and likely nanoplastics are everywhere, everywhere we look. And so just to put this out there, there is a special issue of Applied Spectroscopy that came out last September that has publications from a working group on all of these different aspects of QA/QC, sampling design. There's a paper from Amy Lusher's group on digestion. There's a paper from Sebastian Primke's group on analytical approaches. And there's also a paper on some policy and regulatory developments that are underway in the state of California. So highly recommend this issue. If you can't access it, I can make it available to anyone who has listened in today. It was great to chat with all of you. And again, if you have any questions, I realize I can't be there in person, but if you have questions about anything that was presented today, I'm happy to have an email sent, since obviously I'm not in the room with you right now. But are there any specialized QA/QC practices that you employ in your work that we didn't cover? Any questions about something I said about field sampling, environmental sampling, lab protocols, chemical ID, just let me know. And I'm going to give a quick plug here for the Microplastics Interest Group. This is a group that I've been a co-chair of for a couple of years now. It's a pretty large group, and our mission is to support the advancement of scientific research on plastic particles of various sizes, now found in every part of the environment. And it provides a platform for collaboration on research and management of plastic particles in the environment. We also sometimes have the opportunity to put on special meetings. And actually this morning we finished up a special symposium that was mostly based and mostly featured European researchers, but was a multi-week, 2-day per week online symposium that covered everything from analysis and experimental design and toxicology of microplastics and nanoplastics, to the last session today, which discussed ongoing work to look at the potential effects of microplastics on human health and exposure considerations. And so just a lot of different opportunities to get involved and to network. It's open to students too. And this list of the steering